All right, boys. So this is our new chapter, photosynthesis, right? Photosynthesis. Okay. Before entering to photosynthesis, we should know about different types of organism on the basis of energy uptake or taking of food. So we have three types of organisms. The first one is Autotrophs. Second one we have heterotrophs. Okay, and the third one we have chemoautotrophs. Right? Now, first one we have autotroph, okay, organism that prepare own food. Those organisms that prepare their own food called autotrophs, right? Example of this, we have the most important example that we have plants. Some photosynthetic bacteria also include in algae, also include in this autotrophs, right? They can prepare their own food from sunlight. While heterotrophs that can't prepare food. Example of heterotroph is human, animals. We can't prepare our own food, so we depend on what plants for the food. For the uptake of food, we depend on whom? On plants, right? Chemoautotroph, this is a unique category. In this category, those organisms are involved that are taking their nutrients for the chemicals, right? Okay. They don't depend on light, not like plant or not like, photosynthetic bacteria or algae. They don't depend on sunlight. They are autotroph, but they don't depend on sunlight. Okay. Now, but what they use chemical energy. They use what? They depend on chemical energy. They depend on chemical energy. For example, nitrifying bacteria. Nitrifying bacteria and nitrogen cycle. These bacteria are converting what? Ammonia into. These bacteria converting ammonia into what? Ammonia. Formula for ammonia NH3. Okay. N2, right? Nitrite. That is NO2. Okay. NO2, right? And nitrate. NO3. Am I clear? So the oxidation of ammonia, these organisms are getting energy by what? Through oxidation of ammonia. And when oxidation of ammonia happens, so what will happen? Like oxygen will add, definitely nitrogen will remove, and that will give what? Energy to these organisms. So chemoautotrophs receiving energy from chemicals. Now come toward autotrophs. Autotrophs are what? Example plants. You know, why plants prepare their own food? Because a process happens called what? Photosynthesis. And plants. This process of photosynthesis happens through a specific organelle, and the name of that organelle is called what? Chloroplast. And that chloroplast is present in plants. That is present in plant cell. Now let's suppose this is a plant cell. Okay, let's suppose. Okay, surrounded by a cell wall. Okay, inside the plant cell, this is a vacuole. Am I clear? And this is the nucleus of that. So inside the plant cell, you will see some green things. That is actually what? Chloroplast. So let me take this chloroplast out and check it, study it outside. When I got this chloroplast out, so what I see that, you know, this is actually two membrane structure. One is outer and this one is inner membrane. Okay. Now this region, you can check here, inside this chloroplast there are some sacs like structure. That sac like structure called what? Grana. Singular is granum, okay, while plural is grana. Okay, right? And this region that covered the grana, that region is actually called what? Stroma. Stroma. Okay, now you know chloroplast having own ribosome, 70 is ribosome. That's why they can produce their own photosynthesis, own protein for the process of photosynthesis. Okay, ribosome. Ribosome function is protein synthesis, right? And this also contain what? This also contain circular DNA. DNA and the circular DNA. And I discussed with you already that circular DNA is that DNA whose last two ends are joined one again with each other. Okay. Now this outer membrane, this one is outer membrane of chloroplast. Outer membrane of chloroplast, and this one is inner membrane of chloroplast, right? Inner membrane of chloroplast. These two combine what? Chloroplast envelope. These two actually form what? Chloroplast envelope. Am I clear? Dear students, the topic of discussion for today is photosynthesis. Okay. So today we are going to discuss about photosynthesis in detail and steps involved in photosynthesis. Okay, so the first one we have, you know, photosynthesis. Actually, you know, the photosynthesis the process through which green plants prepare their own food. Photosynthesis. Okay, it's the process through which green plants convert sunlight into glucose, right? The process of photosynthesis will complete in two steps. First is light reaction of photosynthesis, steps that are involved in process of photosynthesis. First one we have light reaction of photosynthesis. Light reaction of photosynthesis, okay. Another we have dark reaction of photosynthesis or Kelvin cycle. So what will happen in light reaction of photosynthesis, okay? In light reaction of photosynthesis, actually the light will fall on the grana. Sunlight will fall in grana part of chloroplast. And that light energy will convert into ATP and NADPH. This energy ATP and NADPH will use where it will use for the fixation of carbon dioxide and stroma. And by utilization of ATP and NADPH, right, this carbon dioxide will convert into what? Glucose. Through dark reaction of photosynthesis. Am I clear? Now, if you see here, as we discuss about the structure of chloroplast, you can see here this region of chloroplast. This region of, this is the outer membrane of chloroplast. This is the inner membrane, right? Inside the chloroplast, the sac-like structure, okay, combine and form what? Grana of the chloroplast. So this part is actually grana of chloroplast on which sunlight will fall. And when sunlight fall on this grana, so this grana part contains chlorophyll. And from that electron will get excited and that will help in the formation of what? That will help in the formation of energy, ATP and NADPH. Am I clear up to here? So listen me very carefully. Sunlight will fall on the grana of the chloroplast and that sunlight energy will convert into ATP and NADPH. This ATP and NADPH will utilize in stroma part of chloroplast. This region is stroma. The place where grana present, this is called stroma. So this ATP and NADPH will utilize where within Kelvin cycle are also called dark reaction of photosynthesis. For what? For the fixation of this carbon dioxide. And this carbon dioxide will convert into what? Into sugar. Am I clear? Now, after utilization, that will, you know, ADH and ATP, you know, when it utilized, so this ATP will convert into ADP, okay, and NADPH will convert into what? NADP. So that can go back toward the grana for what? For reduction again. Reduction for the NADP while phosphorylation for ATP. Is it clear? So remember, here will be, you know, the breakdown of water, that is called proteolysis, will also happen, right? And when this reaction complete, it will give you oxygen also. The equation for this photosynthesis, we have first carbon dioxide plus what? Plus water. So this water actually providing electron. Okay, we will do is now in the step of light reaction. So that both combined, they will form what? Glucose plus oxygen. So this is this oxygen, right? I hope up to here this is clear. Now we will move towards the steps that are involved in light reaction. Remember boys, light reaction will complete in 2 steps. Light reaction of photosynthesis, light reaction of photosynthesis will happen where? In grana part of chloroplast. Okay? And light reaction of photosynthesis, actually 2 pathways are there. These are not steps. 2 pathways. One may be in the form of, that is happening usually, that is in the form of cyclic, that is called cyclic photophosphorylation of light reaction, cyclic photophosphorylation. Another we have non-cyclic photophosphorylation. Non-cyclic photophosphorylation. Okay. Now, usually this cyclic photophosphorylation is happening in plant, but in some cases also non-cyclic can happen. Okay. We will do these 2 pathways in detail, that what will happen in this path. Right? Now, light reaction of photosynthesis happen in grana part of chloroplast. So what is, what is the important things that is present in this grana in which this reaction is happening? So the important thing in grana is actually pigment. Grana contain pigment. If I draw you the, the grana structure, so it is like this. Grana is actually, this is in the form of like this, okay, sac-like structure. Now, if I take it, this one part of this grana or the membrane of this grana. So this membrane of grana is called what? Telecoid membrane. Telecoid membrane. Now, this telecoid membrane is actually, if I study it in more detail, so this telecoid membrane is actually lipid bilayer in nature. Lipid bilayer. 2 layers of lipids, right? This is membrane of grana called telecoid membrane. Okay. Now, this telecoid membrane contain what? This contain pigments for absorption of light or for the trapping of light. That pigment present in the form of cluster, right? This pigment present in the form of cluster. That is called, that is like, I will draw it here, that pigment is present in this form within this membrane, within this telecoid membrane. Like this. Okay. In this form, this is present. In this cluster form, this is the complex of protein plus that pigment. So this is called what? This is called photosystem. Photosystem. So it means photosystem is actually the combination of protein plus pigment, and that pigment actually converting sunlight into chemical energy. That is converting sunlight into ATP and NADP. Now, if we study this photosystem, that is the cluster of protein plus what? Plus pigments, color. So it is like this. Check photosystem. Okay. This photosystem, we have two regions. One region is called what? One region is called antenna complex. Okay. While another region called what? Here supposed to be N, right? Antenna complex. While this region is called reaction center. Okay. Now, this reaction center contains specialized pigment called what? Chlorophyll. Chlorophyll A. Okay. While this antenna complex contain a specialized pigment, like they contain keratinite pigment, chlorophyll B, and also other pigment like xanthophyll, etc. Okay. Now, this pigment is actually called accessory pigment. Okay, while this chlorophyll A is the actual pigment called primary pigment. Am I clear? Now, what will happen? Sunlight will fall on, sunlight will fall on this antenna. This is sunlight, okay? Sunlight will fall on antenna complex. It means accessory pigment will absorb the sunlight. Accessory pigment will absorb that light energy. And from that accessory pigments, that will travel in the form of, electromagnetic energy like this. It will move from one pigment to another. And that energy will reach to where? To reaction center, that is chlorophyll A. This chlorophyll A electron will get excited. And it get excited, it will move to higher energy level. Electron will release from this chlorophyll A. Am I clear? Now, when this electron release from this chlorophyll A, so within membrane, for example, this electron released from chlorophyll A, so from this membrane, within membrane there are different proteins. So the first protein that it receive it is called primary electron acceptor. So that will receive by what? Primary electron acceptor. And that will move through a change of it. One will move to Another electron will get by one, then another, it will move in a chain, but I will discuss you later on. Just be here with me up to here, okay? Now, up to here, photo system is clear. But problem is we have now two photo system. Photo system is clear up to here. We have two types of photo system in this membrane. One is called photo system one, and another is called photo system 2. These both have antenna complex and reaction center, but both are different from each other on the basis of this specialized chlorophyll A. Why? Because this contains that chlorophyll A that is absorbing 700 nanometer wavelength of light. While this one is absorbing which wavelength? 780 nanometer, right? Am I clear? I think no, it is 680 nanometer light. Okay? Is absorbing this chlorophyll A. So that's why these both are different from each other. The basic structure, all the thing is same. Right? You know, boys, just like, you know, this photo system, actually, this contains two parts, right? So let's suppose this part with me is, antenna complex, let's suppose. This part with me is antenna complex. That contains accessory pigment. Okay? While this part with me is, which part? This with me is reaction center, right? These both combine from what? Photo system. That is present in Telecoid membrane. These both combine antenna complex and reaction center plus some protein that will form what? Photo system. Now, sunlight will absorb by whom? By antenna complex. Sunlight will absorb by antenna complex. Sunlight will fall on this antenna complex. Okay. Now, when it absorbs that light energy will, in the form of wave, it will reach to reaction center. Reaction center actually contains chlorophyll A. And when that energy reach to reaction center or chlorophyll A, so that will absorb 700, if it depends on which wavelength, which photosystem it is. That wavelength of light it will absorb. When that absorb it, right? So what will happen? Electron from that will get excited. Electron from that will excite. Is it clear up to here? So electron from this will get excited, okay? And that electron will start moving in a chain. There will be, in this membrane, there are different protein acceptor. There are different electron acceptor protein. So this electron will accept by a protein called what? Primary electron acceptor. Primary electron acceptor. So further, I will discuss this, that what will happen with this electron when it is accepted by primary when it accepted by primary electron acceptor. We will discuss that now in this cyclic photophosphorylation and non-cyclic photophosphorylation. Okay, is it clear up to here? Non-cyclic photophosphorylation is one way of light reaction, right? In this, what will happen? Definitely, you know, this non-cyclic photophosphorylation will happen in the Telecoid membrane of grain. Okay, so how it will happen? You know, first of all, sunlight will fall on what? On photosystem 2. As I discussed with you, it will absorb P680 nanometer wavelength of light. Okay, at that time, you know, when sunlight fall on photosystem 2, that will accept by what? By accessory pigments, and after that means it will accept by reaction retina complex, and that light will move to reaction center. And from reaction center, from chlorophyll A, electron will get excited. And when electron get excited, from that place, you know, it will receive by primary electron acceptor, you can see here. And after that, now electron is going from higher energy level to lower energy level. So different protein, electron acceptor protein will accept that electron. It means redox reaction start. When one will oxidize, another will reduce. From one electron carrier protein, that will give to another, right? So the one that accepted, that protein will reduce. Okay, and it is going now in a change, right? Be with me here. You know, at the same time, you know, light will absorb by photosystem 1, and from photosystem 1, electron will get excited, and that electron will move to higher energy level, and that will again accept by primary electron acceptor, and that will also move through change. Okay, now when this electron is moving through this telecoid membrane, okay, what will happen? For hydrogen ions, channels will open, and when channels open, so that channel will pushing hydrogen from stroma. For example, this is your grana, okay, this is your grana, okay, and outside is stroma. So hydrogen ion will start entering to the lumen of grana. Inside the space of the grana is called lumen. To this region, the hydrogen ion will move. Okay, why? Because electron is moving from higher energy level to lower, so that is taking this hydrogen ion from out to in. Okay, just like that we discussed in the chapter of respiration. Okay, now if you see here, when this electron jump from photosystem 1, right, to primary electron acceptor, so you know this photosystem will remain ionized. Ionized means that having charge because it's lost electron. So that gap will fill by the electron that is coming from photosystem 2. That gap will fill by whom? By electron that is coming from photosystem 2. Is it clear? Now this electron that jump from photosystem 1, at last it will reach by, it will final electron acceptor, it will accept by final electron acceptor, that is NADP. It means NADP will reduce to NADPH in this case. Am I clear? Now come toward this point. When this is pushing hydrogen from out to in, right? So this hydrogen ion will go back from the lumen to stroma through ATP synthase enzyme. And when it is moving back through ATP synthase enzyme, so at that time, ADP will bind with phosphate, so phosphorylation will happen, and ADP will convert into ATP. So here phosphorylation happen with the help of light, right? It means like hydrogen take it from electron, get excited in this reaction by light. So we call this phosphorylation, but photophosphorylation. Okay? While if you check here, this reaction is in the form of Z, called Z scheme, this also called, are non-cyclic, because you know this happen, the electron get excited and go at the end, it received by NAD. So it means the electron did not come back toward its position. It's, you know, it's except by NADP. So the electron doesn't come back toward that photosystem 2, but it's just except by that NADP. That's why it is non-cyclic. Now, one question here remaining with us is that, you know, From photosystem 2 when electron, for photosystem 1 when electron get excited, so that gap fell by electron that was coming from photosystem 2. But what will happen with the spare, because photosystem 1 lost electron that is now ionized, chlorophyll A is now ionized with having charge. So that gap will fill by the lysis of water molecule through sunlight, right? So in the presence of sunlight, breakdown of water will take place in that will release electron. So that electron will fill that gap. Now this reaction is called photolysis. This breakdown of water in the presence of light called photolysis. Am I clear? Now, another question we have, like, the first one coming with us is photosystem 2, right? We supposed to give photosystem 1 name, but no, it is photosystem 2 because of the discovery. It means the first one was discovered photosystem 1. That's why they give the name to that photosystem 1, and to this they give the name photosystem 2. Is it clear? Now the ATP and this NADPH that produce, so this will move to where? And stroma, it will move forward for dark reaction of photosynthesis. It means in the stroma, it will fix, it will utilize in the for the fixation of carbon dioxide. Am I clear? Now our next, this is non-cyclic photophosphorylation. Sometime plants also following cyclic photophosphorylation. So all right. Now, what is the difference between cyclic photophosphorylation and non-cyclic photophosphorylation? Cyclic photophosphorylation only photosystem 1 involved. Non-cyclic photosystem 1 and 2 both involved. Cyclic photophosphorylation, no photolysis of water happen because the electron come back toward the photosystem 1. While in non-cyclic, photolysis of water will take place. Cyclic photophosphorylation, oxygen not evolved. While here in non-cyclic, oxygen evolved because of the photolysis of water. Cyclic NADPH plus H is not synthesized. Means like simply NADPH will not produce. Reduced NADP will not synthesized. Why? Because only, that because the final electron acceptor is again photosystem 1. So in this case, the final electron acceptor we have what? NADP. So it means here NADPH produce. Simply in both ATP produce in both, but in cyclic no NADPH produce. NADPH. While here in non-cyclic NADPH synthesized. Why? Because NADP was the final electron acceptor in non-cyclic photophosphorylation. In cyclic photophosphorylation, electron is cycled back to a reaction center. It means electron accepted back by photosystem 1. Non-cyclic photophosphorylation, electron doesn't return to the reaction center. If they doesn't return to the reaction center, so where that went? That received by NADP. And that NADP convert into NADPH. So the final electron acceptor was in non-cyclic photophosphorylation was NADP. While in cyclic photophosphorylation, electron is cycled and that came back toward the photosystem 1 from where it was excited. All right, so dear students, today we are going to discuss about two important topics. One, we have Hall reaction, okay. Okay, and another one we will discuss about dark reaction of photosynthesis today. These two are the topic of discussion for today. You know, in 1939, Robert Hall, he was a scientist. In 1939, Robert Hall studied this reaction called Hall reaction, and what he proved that at that time, that if I write to you here equation for photosynthesis, so for photosynthesis we need what? We need carbon dioxide, right? Water plus carbon dioxide, okay? It will give us what? Glucose C6H12O6 plus oxygen. Am I clear? What this scientist proved at that time? He proved that these two steps, if you check here, we have two steps here, right? One, we have this step, in which water will give us oxygen, right? This step I have, right? Another step I have, in which carbon dioxide convert into what? Glucose, in which carbon dioxide will convert into glucose, this step. Am I clear? So this Robert Hall proved that these two steps are independent. These two steps are independent step of photosynthesis, right? How he proved that? He isolated what? Chloroplast. He isolated chloroplast, okay, and he isolated chloroplast and illuminated with light. In the presence of illuminated this chloroplast, in the presence of light. It illuminated it in the presence of iron. Right? When he illuminated this chloroplast in the presence of also water. Right? Water. Chloroplast water plus electron acceptor. Plus what? Electron acceptor. At that time, he used iron, electron acceptor. Right? What he found that this water convert into what? Hydrogen plus electron plus oxygen. Am I clear? In this electron accepted by what? By electron acceptor. This electron, right, plus this hydrogen ion accepted by that electron acceptor, that he used iron at that time. Right? And that iron reduced. Correct? when it accepts this electron from water, so that's reduced, iron reduced. Okay, so he found that the conversion of water and to oxygen, it happened in the presence of light and chloroplast and electron acceptor, and that electron will reduce, that electron acceptor will reduce. At that time, he used iron as electron acceptor. And he found that iron reduced by accepting hydrogen plus electron. Right? So he found that this carbon dioxide, conversion of this carbon dioxide into glucose, this is actually the dark reaction of photosynthesis, and this reaction having no link with this light reaction. It means that this, what he deducted from it, what he concluded. He concluded that this chloroplast having the ability, or chlorophyll, right? He isolated chlorophyll, the green pigment. That green pigment having the ability to do what? To isolate oxygen from water and reduce electron acceptance. Okay, so we prove that if we perform experiment, and we can prove this now by using a dye, called what? The name of the dye is called DCPIP. We can perform this experiment by using a dye, and that dye will show us DCPIP. Am I clear up to here? Right? So dichlorophenol, endophenol. This is the short, this is the name for DCPIP. So you know this DCPIP blue in color, right? When it accepts electron, when it reduced, accept means electron accepting means when it reduced. When DCPIP reduced, it will become colorless. So I can perform this whole reaction, and I can prove that. For if I select what? If I select first, if I isolate what? Chlorophyll, okay? In that chlorophyll, I illuminate it with the light, okay? If that chlorophyll, I illuminated that with light, okay? And in the presence of what? DCPIP, in the presence of DCPIP, okay, plus water. So what will happen? This DCPIP is now blue in color, okay? When I expose that solution to light, so photolysis of this will happen, right? And when photolysis of that happen, so water will release what? Oxygen. Is it okay? This hydrogen and electron will accept by what? By DCPIP. And DCPIP will reduce. When this DCPIP reduce, so this will become what? Colorless. When DCPIP reduce, so in this case, it will be colorless. Right? So if my solution that is under the study, if it become colorless, so it show me that the light reaction of photosynthesis take place here, right? If the color is changing quickly, it will show me the rate of light reaction of photosynthesis is high here in this wavelength of light, in this light intensity. Okay? Is it clear? Now, we know already that, DCPIP actually we use instead of the actual reaction in photosynthesis electron acceptor is what? NADP. And that NADP will accept electron and that will convert into NADPH. Right? Robert Hell used what iron at that time is electron acceptor. But we use what? DCPIP dye because it show us the result easily. If it converts into colorless, it show that the reaction took place. Okay? Right? So this NADPH in photosynthesis, this will move toward where? This will move toward dark reaction. This will move toward dark reaction of photosynthesis. This will move toward dark reaction. And in dark reaction, this will utilize for the fixation of what? Carbon dioxide. In dark reaction, reduction of this carbon dioxide will take place and it will convert into what? C6H12O6. Am I clear? So it means this NADPH from light reaction, it will move to dark reaction. Am I clear up to here? All right, boys. So investigating the Hell reaction, if you check the graph, in this graph, we are going to study what we are going to study. Like we are through this graph, we can also find which wavelength are, through which wavelength of light or light intensity chloroplast activity is more, right? So this one side, y-axis of the graph show us if the color of the solution is decreasing, right? Colorimeter, actually, this colorimeter is measuring that color, right? So up here, initially the solution at that was blue, right? And when the light reaction is happening and DCPIP is accepting electron, so that solution is going to become colorless. Okay, so first of all, we isolated what that chloroplast from leafy plant by liquidizing the leaves and ice cold buffer, so that ice cold buffer we are using because the high temperature can damage that chloroplast, and then we will the filtering or centrifuging the resulting suspension to remove unwanted debris, working quickly and using child glassware small tube of buffer chloroplast suspension with added DCPIP buffer we are using to to maintain the pH and temperature we are not increasing so that it may not damage our chloroplast, right? Or the color, the green pigment chlorophyll may not damage. Okay, so then we are keeping that tube at different light intensity, placed in different light intensity or different wavelength of light. In the blue color assess its interval, and then we are checking that blue current at different time interval. So the rate of loss of blue color, right, is measured in a colorimeter, or we can match it with a tube against any known concentration of the DCPIP, or we are measuring through colorimeter, right? It after different time interval, like up to 2 minutes, 3 minutes, 4 minutes, like that. Okay, now First, if you check here, when our sample was in light, so at this point it was blue at this time initially, and then it, the blue color little bit disappear with the sample was in light, right? And gradually it is disappearing at 2, and it below 0.2 it after 5 minutes. Am I clear? So it means that chloroplasts in light that are going to make the dye dissipate colorless because of reducing that dye. Okay. Now, another side of the graph, chloroplast in dark for 5 minutes, then in light. Right? This chloroplast or this solution, we keep it for 5 minutes in dark. If you check here, there is no light, so it's the color remains same. No change happened to that. No reduction of DCPIP happened. It means that the chloroplast needs light. And when we on the light again, so what will happen? The graph or the DCPIP start to become colorless again. Means the reduction of DCPIP start once again. Am I clear? So you can check by using this experiment different light intensity or wavelength of light that which the activity of chloroplast are more. Up to here we discussed about Hall reaction and light reaction of photosynthesis. Now our next topic is the light independent reaction of photosynthesis. Light independent reaction of photosynthesis is, actually this reaction happening without light. This is an independent step from the light reaction. In light reaction, we discussed that sunlight will fall in this grain of chloroplast, and that will convert that light to chemical energy, and that energy with us is what? That will be NADPH and ATP, and that will utilize and stroma part of chloroplast for the fixation of carbon dioxide to convert that carbon dioxide to sugar. So the light independent reaction or dark reaction of photosynthesis are a chemical reaction that convert carbon dioxide into glucose. The reaction occur in the stroma. Stroma is this outer region from the grana. This, you can see this liquid part of the chloroplast, right? Where what is present in stroma, you have grana, the fluid filled area of chloroplast outside the telecoid membrane. Outside the grana means, for example, this telecoid membrane of grana. So this outer region is called stroma. These involve the reduction of carbon dioxide using reduced NADP. It means reduced NADP, we will use it in reduction of carbon dioxide will happen. Carbon dioxide will accept electron and nitrogen from reduced NADP. And ATP, right? And ATP produced in light reaction of photosynthesis. And also in this reaction, ATP that produced in light reaction of photosynthesis, that will convert into ADP. The reaction are known as Cal1, and they take place in the stroma of chloroplast. This is Cal1. This is also called dark reaction of photosynthesis because it doesn't depend directly on light. It's called Cal1 cycle because of the scientists who study for the first time. And this is also called light independent reaction because it doesn't depend on light. Okay. Now let's study this in detail. So, this cycle will complete in 3 steps. There are 3 steps of this dark reaction of photosynthesis. The first one is fixation of carbon dioxide. Carbon dioxide will enter to the stroma, right? And when it enters to stroma, it will react with what? It will react with ribulose biphosphate in the presence of Rubesco enzyme. Ribulose biphosphate carboxylase enzyme. Okay, so in the presence of that enzyme, it will convert into 3-phosphoglycerate. Second, reduction phase of this cycle, in which reduction of substrate will take place. And third one, regeneration of RUBP, ribulose biphosphate regeneration. Of RUBP. This RUBP will regenerate. Okay, so let's see here. If you check here, this one, first of all, this carbon dioxide enter to stroma. In the presence of this enzyme, this is very important enzyme. Remember, ribulose biphosphate carboxylase. This is abundant enzyme because it's present in all the plants, right? In this RUBP, ribulose biphosphate carboxylase enzyme will fix this carbon dioxide, 3-carbon carbon dioxide molecule enter to the stroma, and that react with what? 1, 2, 3, 4, 5. 5-carbon molecule called ribulose biphosphate, and that make what? 3-phosphoglycerate. 3-phosphoglycerate because phosphate is position 3, but the molecule is 3-carbon molecule, right? 3-carbon molecule. It means total molecule we have how much? 6. 6 molecule we have. 6 multiply 3, right or no? Each molecule having how many carbon? 3. 1, 2, and 3. Am I clear? Then that 3-phosphoglycerate convert into 1,3-biphosphoglycerate. Why? Because here the ATPs that came from light reaction that utilize here and that convert into ADP. Am I clear? Then 1,3-biphosphate phosphoglycerate convert into what? That convert into glyceraldehyde 3-phosphate. Still we having six molecule. But here, if you check here, NADP gives electron, right, to 1,3-biphosphoglycerate. It means the substrate here become reduced because it accepts electron from NADPH, right? And NADPH convert back to NADP. Am I clear? So in this case, if you check here in this first phase, when carbon dioxide reacts with ribulose biphosphate, RUBP, at this time fixation of carbon dioxide. Now, this is the phase of reduction in which substrate accept electron from NADPH. Means substrate reduce, 1,3-biphosphoglycerate reduced. So this phase is called reduction phase of dark reaction. And this is important phase. This will now convert into what? You can check it here. Glyceraldehyde 3-phosphate. So 5 glyceraldehyde 3-phosphate, 5 molecule of glyceraldehyde 3-phosphate will make what? Ribulose biphosphate. 3 molecule of ribulose biphosphate. Check here, just count the number of carbon here. 5 molecule we have and 3 carbon, 1, 2, 3. So 5 multiply 3, 15, right? And this can 5 multiply 3, okay, 15, 1, 2, 3, 4, 5, right? 5 carbon molecule and total molecule we have 3. So 5 multiply 3, you got 18 again carbon. Am I clear? So this step in which regeneration of RUBP takes place, because this RUBP will be used for another cycle of dark reaction of photosynthesis. So this is the step of regeneration of RUBP. But here, if you check here, this one glyceraldehyde 3-phosphate, this carbon we got. this carbon backbone we got, and this carbon backbone can use for making glucose or other organic compound in your body. For example, another like this 3 carbon molecule come from another reaction and both combine, so they can make what for you? Glucose. Am I clear to up to here? So we got what from that carbon dioxide? We got that 3 carbon dioxide is now in the form of organic molecule. Now, okay. I hope up to here this is clear all. So boys, this enzyme is very important, ribulose biphosphate carboxylase, because it is combining carbon dioxide with RUBP. Okay, you can check here. RUBP. Ribulose biphosphate plus carbon dioxide in the presence of ribulose biphosphate carboxylase, and it will form what? Phosphoglycerate. Is it clear? Dear students, the topic of discussion is factors affecting rate of photosynthesis. What are the factors that will affect the rate of photosynthesis? Okay, now we know already that photosynthesis needs different factors. So let's see, for example, this is the equation for photosynthesis, and photosynthesis need what? Carbon dioxide, water, right? It will form what? C6H12 and O6 glucose, right? Plus oxygen. This will be the product, right? Now this reaction need what? The first thing is for the light reaction. We know for this light reaction, that need what? Light, sunlight. Okay, first is that sunlight. One factor is that light intensity is very important for the light reaction of photosynthesis, right? Light intensity is very important for the excitation of electron from chlorophyll. So one factor we have light. Another, for dark reaction of photosynthesis, we need what? Carbon dioxide to convert that into glucose. So carbon dioxide is another important factor that we need, right? So this carbon dioxide is also one of the another factor that need for the photosynthesis. Another, we know already that the process of photosynthesis and process of photosynthesis, several enzymes involved, right? Like one example I told you already, ribulose biphosphate carboxylase, that's involved in dark reaction for the fixation of carbon dioxide. And we know already that for enzyme need what? Enzymes need optimum temperature. Enzyme working at optimum temperature, right? So temperature, sunlight, and carbon dioxide. These are the important factors that need for photosynthesis. Temperature need for the enzyme activity, because if you are decreasing the temperature, enzyme will not work well because the kinetic energy of the molecule is less. right? So to work better, enzyme need optimum temperature. So one of the factor for photosynthesis, we have temperature. So this one we have temperature, this one sunlight, and carbon dioxide. These are three main factors that can limit if the deficiency are, it means like if these factors are less, if these factors are restricted, so that can decrease or that can minimize the rate of photosynthesis. Photosynthesis will not be at that rate which supposed to be. Any factor that are restricted in this, that and due to which rate of photosynthesis is not more or not that reach to maximum, so that factor is called limiting factor. So let's suppose first one we will do light intensity. Right? Light intensity, sunlight, if it is, if the sunlight having no more intensity, right? And that is, like it's not much intense, so like it may not bright, so that can affect your rate of photosynthesis. Because the sunlight needed for what? Our light intensity need for electron excitation from chlorophyll. Right? And light reaction of photosynthesis. This electron excitation will happen from chlorophyll. Right? Now, if sunlight is, light intensity is important, but it should not be at very high range that it may damage the cell. Now, it should not be like this. Here I'm talking about the specific intensity that need for the electron excitation from chlorophyll. Now, if I draw a graph from this light intensity, so you can see here, graph will be like this. One side I will mention what's light intensity. Right? Another side, rate of reaction or rate of photosynthesis. Right? Now, if you are drawing the graph for it, so this graph will become like this. If you see here, when light intensity is increasing here, you check, so rate of photosynthesis is increasing. Am I clear? But a time will come with you that this graph becomes flat. It becomes straight. Why? Still I'm increasing the light intensity and no effect of that on rate of photosynthesis. So what is the reason behind this? Is, here, initially, boy, if you see here in this place, I'm increasing light intensity, so rate of reaction is increasing. So in this place, my rate limiting factor was what at this point, let's suppose. Rate limiting factor at this point is light intensity. When I'm increasing rate of photosynthesis increase. So why it is less in this point, it means light intensity is less. But when I'm increasing rate photosynthesis reach to that maximum. But now I am up to this point when I'm increasing, so there is no effect of that light intensity on rate of photosynthesis. So it means at this point, maybe rate limiting factor may be carbon dioxide, maybe no enough carbon dioxide, maybe no enough chloroplast. because chloroplast need number of chloroplast also depend rate of photosynthesis. Is it clear? Maybe that may be temperature. That's why my reaction rate is not increasing. Am I clear? So rate limiting factor, then when the graph becomes flat, so maybe it's maybe this, or this. Okay, so let's do this experiment and a scientist prove that that's how it can affect. And first I will tell you about the effect of carbon dioxide, and then we will merge both for light intensity and carbon dioxide. I will make another graph in which I will check these both factors. Okay, so let's suppose now I am considering carbon dioxide concentration. What? CO2. This is another factor for photosynthesis. One side carbon dioxide concentration, right? And this is the rate of reaction. Remember, boys, this carbon dioxide concentration is under your control. Means you can increase it by your will during experiment. So this variable is called independent variable. We keep this independent variable always on x-axis. While rate of reaction is dependent because it depends on this carbon dioxide concentration, H will be y-axis. Now for the carbon dioxide, you will also see graph just like for the light. You can see here, is carbon dioxide concentration increasing, so rate of photosynthesis is increasing, right? But time will come that this become plateau. So it means initially in this range, carbon dioxide was the rate limiting factor. But when it reached to plateau, so it means the rate limiting factor may be light, maybe what? Maybe chloroplast, other any factors can be, due to which photosynthesis is not increasing anymore. Even I am increasing the carbon dioxide concentration, but no effect of that on rate of. So maybe there are something other like light chloroplast, due to which this reaction is restricting, due to which that are restricting the rate of photosynthesis. And we know that are minimizing or restricting your rate of reaction, that are called what? Rate limiting factor. Now, let's do for temperature. Then we will do a merge graph. Another factor we have what? We have temperature. Right? Let's see. One side, you know, temperature, again, it is under your control. So you will mention temperature here, x-axis. All right. And y-axis, write it here, rate of reaction, or rate of photosynthesis. Now, For temperature, the graph will be not same just like we did it. For temperature, you will see the graph just like check it here. Now, if you see here, when I'm increasing temperature, rate of reaction is increasing up to here. It means that at this point, at this temperature, rate of reaction is high. It means this is optimum temperature for the enzymes that are involved in the process of photosynthesis. But above that, when I'm going, my rate of reaction is going to become decrease, even it reaches zero. Why? Because initially when I was increasing temperature, so that temperature was increasing the activity of enzyme, that was increasing the kinetic energy of the enzyme and substrate, so there were more collision and the rate of reaction gradually increasing. More collision between enzyme and substrate. Enzyme was able to convert that substrate into product. At this point, it was the optimum. At this maximum, substrate was changing into product. This was the optimum temperature, the most favorable temperature for enzyme. But above that, the enzymes start to denature. Enzymes are proteinaceous in nature, and if you are increasing the temperature, that enzyme will damage. And we call that denaturation of enzyme will take place. Denaturation of enzyme will take place. Right? Now, let's see, we are going to prove that limiting factors having effect on rate of photosynthesis. The first experiment, I'm considering two factors. One, I'm considering carbon dioxide concentration, right? Another one, we have light intensity. Okay. Let's suppose I have a graph for it. One side mention what? Light intensity. Another rate of reaction. Rate of reaction means rate of photosynthesis. Okay, now I will design 3 experiments. One experiment, I will consider what? 0.04% carbon dioxide. Am I clear? Another experiment, I will consider 0.4%. One is 0.04, another 0.04% carbon dioxide. Okay, these two I will consider with that. Now, first, when I perform this experiment, right, when I perform this experiment with, the light intensity, I will keep it here, definitely, the light intensity here, is it increasing? So, sorry, actually it is increasing, so it means up to here, but it graph becomes flat. Here I used what? 0.04% carbon dioxide in this first experiment. right? So what I figure out from this here, that when I was increasing my light intensity, rate of reaction was increasing. But a point comes that the graph becomes flat to no more effect of this light intensity. So what I did in second experiment, the same experiment I performed with 0.4% carbon dioxide. We can increase or decreasing carbon dioxide concentration in our solution by using sodium hydrogen carbonate. Okay, so 0.4% carbon dioxide, I use that, so the rate of reaction go up and then it become again flat. So it means that in this case, in this graph, at this point, like the one experiment that I performed for the first time, rate of limiting factor was what? Carbon dioxide, due to which that graph go up, right? Is it clear up to here? Now, another experiment you can also perform and you can check the effect of temperature also. For these two experiments, for this first one, let's suppose I keep the temperature, I kept the temperature 15 degrees Celsius. Now I'm comparing another factor. And when I move towards 0.4%, right? When I move towards 0.4%, at that time I kept the temperature 25%. So it means that rate limiting factor was what? Temperature and carbon dioxide. Is it clear? Due to which your graph become high. Another experiment, what I did, I keep this 0.4 constant. I perform another experiment. This 0.4 I keep. I just want to check it now. That at this point, what is the rate limiting factor with me? So, okay, sorry, boys, please. Here, this experiment with me is 0.04. Okay, another also 0.04. Okay, boys, just please make it correction here. Now I'm modifying this 0.04. Okay, this one I also kept it 0.04. I kept this bar also 0. But only I increase temperature. When I increase the temperature here, right? So what happened? When I increase the temperature from 15 to 25, my graph go up. So it means at this point, the rate limiting factor was what? Rate limiting factor was temperature. Even I didn't change carbon dioxide concentration, but still the graph go up. So rate limiting factor at this time was what? Temperature. Listen me very carefully. Here, I keep these two what? Same, carbon dioxide concentration. I didn't change that. Only I changed the temperature, so my graph become up. That go up. And third experiment, what I did, I increase that carbon dioxide concentration and I keep that 0.4%. And the temperature remains same. Temperature remain same. Temperature remains at 25, but the graph go up. Still the graph go up. Why? Because of increasing what? Because of increasing carbon dioxide. So in this graph, in this place, in this rate limiting factor was what? Carbon dioxide. That's why it becomes flat. At this point, rate limiting factor was what? Temperature. Am I clear? And at this point, the photosynthesis is at maximum. If it is plateau, so maybe because of the number of chloroplasts. So listen me very carefully. At this point, rate of limiting factor temperature. Right? So at this point, we have what? rate limiting factor was what? Temperature. When I increase the temperature, my graph go up. Even I keep the, what you carbon dioxide thing. And then here when I kept temperature same, but increase the carbon dioxide concentration, so the graph go up. So it means at this point, rate limiting factor was what? Carbon dioxide. At this point, rate limiting factor was temperature. Am I clear? And here the graph is at maximum. Maybe this become plateau. Maybe the maybe because of the chloroplast number. That's why the graph again become plateau. I hope up to here this is clear. If you do have any question, you can ask for. All right, boys. So today the topic of discussion is investigating rate of photosynthesis using an aquatic plant. So here we will check three parameters, right? At which parameter or at which factor the rate of photosynthesis is high, right? So even we will also check another factor, that is the wavelength of light. So the first one, I will write to you here, the first one parameter or the first one factor that we are going to study. This one we have the first one we have what? Light intensity. Light intensity. Okay. Second one we have wavelength of light. Okay, third one we have concentration of carbon dioxide. Okay, and fourth one we have temperature. So if we are increasing or decreasing these factors, that having effect on rate of photosynthesis, right? Because light intensity is very important for the excitation of electron from chlorophyll or from primary pigment. Okay, during light reaction of photosynthesis. Wavelength of light is also very important because photosystem 1 and 2 are observing different or specific wavelength of light. So that is also very important. So we should know at which wavelength of light photosynthesis work at a maximum rate. Concentration of carbon dioxide also very important because carbon dioxide need for to convert for the formation of glucose and dark reaction of photosynthesis. Carbon dioxide react with the ribose biphosphate to form what? To make any organic, in dark reaction, it will make any carbon skeleton. It will form carbon skeleton that can later on convert into glucose, right? And glucose or any other organic molecule that can convert. So carbon dioxide need for that. Temperature also needs for enzymes. Temperature is very important for enzymes because in the process of photosynthesis, there are many enzymes involved. So for that, we know that enzyme need optimum temperature. So optimum temperature is also one of the important factor, very important for the photosynthesis. So now we having this set of apparatus. Through this we can check all these parameters or these factors. So the first one you can check it here. How we will know We selected in a Canadian pond wheat or any other aquatic plant you can select, right? So here, this is why you take what water, because you know water we need it for the photosynthesis. So here this is water, okay, and water also need for plant. And here, you know, this is green leaves. This is photosynthetic plant. So if photosynthesis is happening in this plant, so is a result of that, what will produce? Oxygen will produce, because we know that in photosynthesis in light reaction, lysis of or photolysis of water will take place, and that will produce oxygen is a waste product. Now this oxygen when coming out, it will produce bubble and water. So if bubble are, you know, if bubble are more in water, right? These bubbles are increasing, are more in water. So this is the indication of oxygen production. It means oxygen is producing. If it is more, there are more bubbles, so it means more oxygen is produced. And that is the indication that rate of photosynthesis is high. Or if it photosynthesis. Now, if we have less bubble, it means that rate of photosynthesis. By counting these bubbles, we know that, you know, that we can analyze, or we know that easily that how photosynthesis is going. It is increasing or it is decreasing from this number of bubbles by counting these bubbles. But it is a very difficult to count these bubbles. So for that reason, we connected this up, this setup with gas syringe. Okay, so this syringe actually, you know, we connected this syringe through pipe, right? With this setup, right? So you can see here, if oxygen production is increasing, so definitely that oxygen will move from here and will enter to this gas syringe. Now, if this gas syringe is moving back, you know, this piston of this syringe is going back. So it means this, in this space, oxygen is accumulating, right? You are collecting that gas oxygen here. So if the pressure of oxygen is increasing, so this piston will grow gradually back. If this cover more a place, means you have to measure this distance. So for example, in one experiment, it covered that distance from here to here, right? Another experiment, it covered less space. So it means the rate of photosynthesis is less in this experiment number 2. Why? Because this piston it covered less distance, less oxygen accumulate here. So it means rate of photosynthesis is low. Am I clear? Now, you can check here. This is one side, this show you volume of gas, right? Volume of gas per ml or centimeter cube. So how much volume of gas you collected? So you will mention that in y-axis. And how here you can mention the time, that how much time it takes, right? So initially you can see here reaction slowing. You know, initially this reaction is going to reach to, it's increasing, right? At this point it is slow, right? And you know, but gradually you can see gradually reaction is increasing, and at this time your graph become flat, right? So if this become flat, which means here reaction approximately stop here, right? So measuring the initial rate of reaction per minute, okay? So you can measure from this graph at different point, that at which point the rate of reaction is high. So if you see here, the graph is going more steepy. The graph is going more steepy means that at this point rate of reaction is high. So you can find it, right, at different point, that initially how was it and now how was it. So at this point, the reaction slow down is compared to initial, right? So initially rate of reaction is high. So obviously more availability of enzymes and chloroplast, okay, and then it gradually decreases, right? So through this way you can measure. So now we are checking these factors. For example, I want to check light intensity, effect of light intensity on rate of photosynthesis. So what I will do, I can, I will expose this apparatus to what? To light, to different intensity of light. For example, I keep it here, source of light like lamp here from this distance. Am I clear? We know already that light intensity is actually uh, inversely proportion to what uh, distance. Is it clear? So the formula for that we have 1 over d square, d represent to what distance. Am I clear? So it means that if you are increasing the distance, so the light intensity will decreasing. If you are increasing the distance, both are inversely proportional. Light intensity is inversely proportional to what? To distance, right? Now, look at here. Here, for example, this distance is, in one experiment, I keep this lamp here, right? Another experiment, I keep it here, right? So here in the second experiment, the distance is more now, right? So this one is for the first and this one for the second. So the distance is increasing, so it means the light intensity is decreasing, because you check here, if distance is increasing, light intensity should be decrease. So here in second experiment, I increase my distance. So when I increase it, so it means the light source is far away from the separator, so light intensity will be less. So you have to measure the distance, okay, and then you will put in this formula, okay? All right, so dear students, the formula for the light intensity we have, light intensity is equal to P is power of light source and 4 phi d square, right? Now d is actually what? D is distance with us. Now let's suppose so here I am considering this distance d square, okay? So light is equal to, light intensity is equal to 1 over d square, means distance square. So what does it mean? So let's suppose if I have a light source, right, and that light source is 1 meter away, right? That is this light source, and I'm considering this distance 1 meter away. I'm considering, so 1 meter away, light intensity is, for example, it is 300 watt per meter square. Am I clear? Now if I'm increasing, let's suppose if I increase this distance and it is now 2 meter, right? So what it should be, what should be light intensity? So I will put this value in formula 1 d square, right? So d square is distance with me, so d here, this is case number 1, this is case number 2, right? For case number 1, light intensity is 300 watt per meter square, but for case number 2, I have, it is 2 meter, so I will keep it here 2, so it will become what? 1 over 4. So what mean by this 1 over 4? It mean
if you increase, if you double the distance from the light source, so that light intensity will decrease by what? By 4. How? Let's see. Initially when it was 1 meter, so light intensity was 300 watt per meter square. But now when I double the distance, so what should happen? What will happen here? The light intensity will be 300 divided by 4. So when you divide 300 by 4, so what you will get? You will get 75 volts per minute meter square. What's 75 volt per meter square? So here, make clear. Now let's suppose if I make my distance what? 3 meter. If I keep if here at this point I am finding light intensity 3 meter away, so that will be what? Again put that value in the formula, so that will become 1 3 square, so it will become 1 over 9, right? So the original was it's 1 meter, it was 300, so now it's 9, it will become what? 300 divided by 9, so it will become with me, 300 divided by 9, so 33.33 watt per meter square. So what we conclude from it, that at 1 meter, when it was the light source, it was 300 watt per meter squared light intensity. When I double the distance, when I double the distance, so light intensity decrease, right? By what? By 4. Now when I increase my distance again, so I got this value 33 point. So from this light intensity decrease how much time? It, in this case, it decrease what? 4 time, right? And now in this case, when I triple the distance, so light intensity decrease by 9, right? So this it, if we are keeping our light source away from the plant, so the rate of photosynthesis is the light intensity will decrease gradually. The light intensity will decrease and rate of photosynthesis will also decrease. We can measure this light intensity. For example, this is our experiment. Here we have plant, right? So from for example, this is plant, this inside this tube we having aquatic plant. So let me make it through another, all right, another color so that you can see. For example, in this tube we have plant, right? So you can keep your light source at different distance. For example, it first you keep it at 1 meter away, then you keep it 2 meter away, and then you can keep your light source 3 meter away. Now when you it each at each distance you will find the rate of photosynthesis. So rate of photosynthesis you can measure through formation of bubble or gas that collected in gas syringe, right? Then you will put that value in this formula, right? That, well, if we put it this value here in this formula, for example, 1 meter, what is it? 1 meter, what is light intensity? So you will put it here 1 meter, and then P is actually which light you are using, what is the watt of your light source. When you put it, that it will show you light intensity for 1 meter, same you put that value here, it will show you for 2 minutes, and same you put that value here, it will show you for 3 meter distance light intensity. So at different light intensity you can find what rate of photosynthesis. I hope this is clear to you all. Now another factor we have wavelength of light. So how we can change it, right? We have, for this wavelength of light, we will use different color filter. For this wavelength of light, we will use different color filter, different filters. We will keep that on the way of light. But remember, the light source should be at the same distance. For example, from here, I'm keeping my light source. From here, I'm keeping my light source, right? So this distance up to here, this should be same for R. For example, one color filter, I keep it here, right? So when I do this experiment in another, I will keep it in the same position and same distance of light source. Okay, so it will, this color filter, so this it will definitely, it's a different color filter, so it means like different wavelength of light will pass through that, and it will show you that at which wavelength, at which color it is, light is working, the rate of photosynthesis is high. Another one we have concentration of carbon dioxide. We can increase or decrease concentration of carbon dioxide. Okay, we can increase or decrease concentration of carbon dioxide. How? Let's see. We are using a special chemical called what? Sodium hydrogen carbonate. Sodium hydrogen carbonate. Sodium hydrogen carbonate. Am I clear? So sodium hydrogen carbonate, NaHCO3. When I'm keeping this in water, so right, it will react and carbon dioxide will eliminate in water. Right? So if I'm increasing this sodium hydrogen carbonate, so it means more and more carbon dioxide will produce in this water. So I can make different setup like this, and I can increase or decrease this concentration of sodium hydrogen carbonate. So for example, in one experiment, I keep it less, another I keep it more. So more sodium hydrogen carbonate. It means that to one experiment or one plant, I expose to more carbon dioxide concentration and to another less. And then I will check how much oxygen I'm collecting in this space, in this gas syringe. It will show me the rate of photosynthesis. The one in which, if the gas collected are this pest and move more, so it means the rate of photosynthesis is high at that concentration of carbon dioxide. Okay, another factor we have temperature, right? So here we can, we can do like, we can sit this apparatus to, like, for example, we want to check the effect of temperature on rate of photosynthesis. So at that time, one setup, I will keep it right at a constant temperature. For example, in the beaker, even I can put water, hot water, but I will make sure that this water temperature should not be changed, right? For example, I'm exposing this to 37 degrees Celsius, right? So this apparatus in this beaker. So this water should not, this 37 should remain constant. Another apparatus I am exposing to 40 or 45 temperature. So this should be remain constant in all experiment. That means it should be remain same. This temperature should not be changed. If it is, for example, if it is decreasing from 37 to 36 or 35, so I should put more hot water so that it remains same. Or even right? So different ways I have that through that, you know, if you can also keep this in water bath at constant temperature, right? But if it is possible that whole setup apparatus, if you can keep that, so water bath can also provide stable environment temperature to your experiment. So you can do like this also. Okay, so it is the effect of temperature on mm, this experiment. But whenever if I'm checking the effect of temperature on this on rate of photosynthesis, I will make sure that other parameters, other factors should remain constant at all. Because only I will change the temperature. I want to check only the effect of temperature. So the light intensity for all the setup or for all experiment that I'm doing for all that, it should be same light intensity and same sodium hydrogen carbonate concentration in R, so that other factor remain same. So then I can get a fair result. Okay. Dear students, the topic of discussion for today is C4 photosynthesis, right? C4, first we should know about C4 plant and C3 plants, right? C3 plants, up to now we discussed those plants, you know, we discussed in Kelvin cycle of photosynthesis or in dark reaction of photosynthesis, in which carbon dioxide will react or it will combine will combine with RUBP, ribulose bisphosphate, and that will form what? Phosphoglycerate, 3-phosphoglycerate. Am I clear? So that 3-phosphoglycerate is actually 3 carbon molecule. Am I clear? So that's why we in those plants in which this dark reaction of photosynthesis happen and form 3 carbon molecule. The first product is 3 carbon molecule. That is called what? That plants are called C3 plants. Okay. Now we have another other plants, you know, some other plants are there that are living at high intensity of light or in hot condition. So that plants having a specific adaptation. We will discuss that adaptation now. Right? So that because of that adaptation, the Kelvin cycle that is happening in that plant, it is in another way. And through Kelvin cycle in that plant, the first product produced, it is what? 4 carbon molecule. You can check this one. First product of Kelvin cycle is what? Oxaloacetate, that is 4 carbon molecule. So that's why these special plants are called what? C4 plant. We will discuss about these plants in detail now, that what will happen in C4 plant and why they are doing this dark reaction of photosynthesis in another way. Am I clear up to here? So up to here, so C4 plants having, like, there are different C4 plants like sugar cane, corn, right? So these are C4 plants in that dark reaction will happen through another, right? Now come toward this. In dark reaction of photosynthesis, we discussed that carbon dioxide will combine with RUBP, ribulose bisphosphate, and that will form phosphoglycerate, 3 carbon molecule, right? In the presence of which enzyme? Ribulose carboxylase. This ribulose carboxylase enzyme is actually fixing carbon dioxide when there is more carbon dioxide concentration. That will fix that and will convert it to phosphoglycerate. But you know, some plants having problem, they are living at
high intensity of light and the rate of light reaction is high in that plant. When the rate of light reaction is high in that plant, so it means oxygen is more. We know that in light reaction through by photolysis of water, oxygen will be. So in light reaction of photosynthesis, oxygen will be. Is oxygen more in light reaction of photosynthesis, right? If in that plant the oxygen production is more, is compared to carbon dioxide, it means like carbon dioxide entry is less while oxygen production are more in that plant. Why? Because light reaction is high in that. They are living at high intensity of light. So at high concentration of oxygen, this ribulose biphosphate enzyme, ribulose biphosphate carboxylase enzyme can't fix carbon dioxide. It will not fix carbon dioxide. What at that time they are fixing oxygen. They are combining oxygen with RUBP and forming what? Phosphoglycerate. But along with phosphoglycerate, they are making another product that is not needed for our body. That is like waste product. That is called phosphoglycolate. So if we no need for a body this phosphoglycolate, so our body is converting that phosphoglycolate back to phosphoglycerate. Because we need this phosphoglycerate for RUBP formation. And RUBP we need for dark reaction. Am I clear? Now if you check here, this oxygen fixed by ribulose biphosphate carboxylase, because in photorespiration, this is called photorespiration, right? Oxygen is more, right? So RUBP is fixing oxygen. This is called photorespiration. Oxygen fixed and that form phosphoglycerate and phosphoglycolate. This phosphoglycerate can make RUBP again. But what happened along with that, phosphoglycolate form. Phosphoglycolate is 2 carbon molecule. Am I clear? So this phosphoglycolate, our body or our cell have to convert it, no, plant have to convert back into phosphoglycolate. When that have to, sorry, I'm using the word our, but I'm talking this about plants, okay, be clear. So when this have to be converted into phosphoglycolate, phosphoglycerate, so on the way, you know, what will happen? Carbon dioxide will release. When phosphoglycolate, if you check here, we have, for example, 6 molecule of phosphoglycolate. And this is 2 carbon molecule, right? So total number of carbon, how much? 6 multiplied 2, 12, right? This will form what? Phosphoglycerate. Phosphoglycerate is what? 3 carbon molecule. So this 6 phosphoglycolate will form how much? 3 phosphoglycerate molecule. It means phosphoglycerate 3 molecule and phosphoglycerate is 3 carbon molecule. So 3 multiply 3, what? 9. 9 carbon we got from that. So it means on the way it released carbon dioxide. So we lost this 3 carbon. When we lost this 3 carbon, so what it means that we are losing what? RUBP. If that reaction is happening normally, if this phosphoglycolate didn't form, so what will happen? Am I clear? If this phosphoglycolate didn't form, so the one thing is RUBP will be more with us. But another thing, when it lost this carbon, right? One side, if you think we are losing RUBP, how we are losing? Let's see. One point is that maybe our RUBP become decreased from normal. But if still RUBP not decrease, so we can face problem of what? No glucose will form. No, this we will not gain this 3-carbon skeleton. If we not get this 3-carbon skeleton, so no glucose will form. Am I clear? Or if this 3-carbon skeleton form, so we may lose what? We may lose RUBP. 6 RUBP may not form. 5 RUBP will form. So it means that if we are getting this carbon skeleton for the glucose formation, so RUBP will be enough. But usually this glucose, if this glucose form with us, usually like some will form glucose. If this glucose forms, so RUBP will decrease. And gradually, if this RUBP is decreasing, so it means that we are losing RUBP, and when we are losing RUBP, the dark reaction will decrease, and when dark reaction of photosynthesis decrease, so what will happen? Glucose formation or organic material for molecules formation will decrease in plants' body. Am I clear? In order to avoid this problem, and in order to avoid himself from photorespiration, in some plants, those plants that are living with high intensity of light and in which oxygen concentration is more, in that plant certain adaptation to avoid that problem of photorespiration. So that adaptation are what? Let us do. First one is these plants, these are called C4 plants. And C4 plants, this is mesophilic cells, right? And this is vascular bundle of that mesophilic cell. A vascular bundle of that plant. Around this vascular bundle, there is what? Bundle sheet. Bundle sheet. Am I clear? And this bundle sheet are attached like this is, you can check here this bundle sheet. These cells are strongly bound, right? Means there is no gap between them. Am I clear? So let's suppose if photosynthesis is happening and these are light reaction is happening in these mesophilic cells because they contain chloroplast. Oxygen will produce, right? So it means oxygen concentration here is more. Oxygen concentration here is more. Am I clear? And if in this mesophilic cell come here, boys, if in this mesophilic cell oxygen is more, right? So what will happen? If here is ribulose biphosphate, that will fix what? That will fix oxygen. But in this plant, there is another enzyme called what? That enzyme is called PIP carboxylase, phosphoenol pyruvate carboxylase. This enzyme is in the mesophilic cell. So when this enzyme is in mesophilic cell, so let's suppose carbon dioxide enter to this mesophilic cell, right? Carbon dioxide enter to this mesophilic cell. That carbon dioxide will fix by what? By this phosphocarboxylase enzyme. Here is no ribulose because in this plant, light reaction is more, oxygen production is more in this plant. That's why in this mesophilic cell we have another enzyme instead of ribulose biphosphate. We have another enzyme, PEP carboxylase. That PEP carboxylase can fix only carbon dioxide. That will not fix what? That will not fix. That will not combine oxygen. So if oxygen is more, no matter. So that PEP carboxylase will combine this carbon dioxide with what? With phosphoenol pyruvate. This is 3-carbon molecule that is present. And and C3 that was ribulose biphosphate. But this is PIP, phosphoenol pyruvate. This PIP will combine with what? With this carbon dioxide. And that will make what? Oxaloacetate. This is 4 carbon molecule form. This 4 carbon molecule, because of this first product of this reaction, right? This is the first reaction, you know, the first product that is happening after fixation of carbon dioxide. Here, in Kilwan cycle fixation of carbon dioxide, the first product that formed it was ribulose biphosphate. It was 3 carbon molecule, glycerol, phosphoglycerate. But here, this first product is oxaloacetate. That's why they are called C4. This oxaloacetate will convert in this mesopelic cell into malate. Malate is 4 carbon molecule again. That malate will enter to where? To this bundle sheet, these cells, to these cells. And when malate enter to these cells, so in this cell, malate will convert into pyruvate. And when malate convert into pyruvate, when it is converting into pyruvate, it will release carbon dioxide. Am I clear? So when it releases carbon dioxide, so inside this bundle sheet, there is RUBP, ribulose biphosphate carboxylase waiting. That RUBP will fix this carbon dioxide with this RUBP carboxylase enzyme will combine this carbon dioxide with RUBP molecule. And again, glycerol, phosphoglycerate will form. And again, normal dark reaction will take place here. It was happening in C3 plant. And it will form sugar, and it will form that sugar will go to vascular bundle. Definitely, it have to be transported through that, through phloem, right? So here, if you check here, boys, actually nothing happened here, but just we transfer carbon dioxide indirectly from higher concentration region of oxygen to the region where there is less or no oxygen. So that in that place, this RUBP or ribulose carboxylase enzyme fixed carbon dioxide. Because this RUBP carboxylase enzyme, if oxygen is more, they can't fix it. And here this mesopelic cell oxygen is more because of high light intensity, because of high light friction of photosynthesis. That's why this PEP carboxylase enzyme help us and take this carbon dioxide indirectly to where? To bundle sheet. So the adaptation in the C4 plant having what? Bundle sheet. Am I clear? This is one adaptation in this bundle sheet. Okay. Another adaptation in these having, you know, these all are in the diagram, these all that's surrounding the, vascular bundle. So I figure out this is mesophyll, but you know, this highly that's attached with each other very tightly. These are what? Bundle sheet. Okay. The outside, whatever is that are was mesophyll, right? And the between this is vascular bundle, means xylem and phloem. Am I clear? So here, what are the adaptation in these C4 plants? The first adaptation is they having what? Bundle sheet. Bundle sheet. Am I clear? Bundle sheet in which this normal Kilwan cycle will take place. Am I clear? And another adaptation in this plant have they having enzyme called PEP carboxylase. So one is bundle sheet. Okay. Another adaptation they having PEP carboxylase enzyme, phosphoenol pyruvate carboxylase. Is it clear? So here, one oxygen, the oxygen concentration is increasing, one because of light direction, right? Another, also because of to plant entering of oxygen is happening for the respiration, right? So these two factors, these two things contribute to high concentration of oxygen. I hope up to here this is clear. Photo respiration. Okay. Listen me very carefully. Photorespiration is the process in which fixation of oxygen happen with what take place with RUBP. RUBP is like, we discussed this in Kelvin cycle, right? And that convert into what? 3-phosphoglycerol glycerate, right? And phosphoglycerate. Plus, right? Plus phosphoglycolate. Plus phosphoglycolate. Now, this phosphoglycolate is 2 carbon molecule. Am I clear? And this is waste product our body, plant body no need for this. Of this. So, this combination of oxygen with RUBP will happen when oxygen concentration. And the presence of enzyme, what ribulose biphosphate or bisphosphate carboxylase enzyme, right? This enzyme normally binding what? Carbon dioxide with RUBP in dark reaction of photosynthesis, right? Ribulose biphosphate carboxylase enzyme. This enzyme, RBC, I just write it, okay? So this enzyme normally in dark reaction of photosynthesis combining carbon dioxide with RUBP to make what? 3-phosphoglycerate. Right? In this 3-phosphoglycerate, right, it will form what? RUBP again. And when it is forming RUBP again, on the way, it will also give us what? 3 carbon skeleton molecule. Why? Because if these carbon are, for example, 3 carbon dioxide, so this 3 carbon dioxide will give us bank. And that will form what? Glucose or any other organic molecule. Am I clear? And this 3-phosphoglycerate, it will form what? Again, RUBP. But what happened to some plants, those plants that are living in hard condition? They are facing problems. Remember here, that's the first product, the first year I'm talking about this. Carbon dioxide plus RUBP. This first one, this form, this is 3 carbon molecule. Who make? By this ribulose biphosphate. This is why these plants are called C3 plants. Normal dark reaction is happening in this plant. Why normal? Because this enzyme having quality, if carbon dioxide is more, and oxygen is this is fixing carbon dioxide. So normally happening like this. But what happened with plants that are living in hard condition? That plant actually at hard condition, they are closing their stomata. And through stomata at daytime, what's supposed to happen? At daytime, oxygen should go out and carbon dioxide should enter to that plant, right? Carbon dioxide for photosynthesis, and photosynthesis, if oxygen produces the result of nitrogen, that will go out. It's happening normally. What will happen, because these plants are living some under hard condition, in order to evaporate, to prevent himself for water evaporation, for evaporation of water, for evaporation of water. These plants, why? Because it is hard and they have to conserve water. They are keeping stomata close. When they are keeping stomata close, so it means carbon dioxide will not enter and oxygen will not go out. It is hard condition, light intensity is also more. It means light reaction is happening at very high rate. So oxygen is producing that will accumulate in what? In this plant part. It means oxygen concentration increase in carbon dioxide. Am I clear? So what will happen? This ribulose biphosphate carboxylase, ribose enzyme, right? Ribulose biphosphate carboxylase. This is, this is very smart enzyme. This enzyme will now start to, that will make friendship with oxygen. It will start to, because oxygen is more here. So it will start to combine oxygen with RUBP. And when it form oxygen to, when it fix it, so it will form glycerol and glycerate. It will form, yes, yeah, phosphoglycerate, right? 3-phosphoglycerate it will form. But along with that, okay, this 3-phosphoglycerate will make what it will make for me, ribose, RUBP again. But it will also make what? Phosphoglycolate. Phosphoglycolate. And that phosphoglycolate is 2-carbon molecule. That phosphoglycolate have to convert back into this phosphoglycerate. When this is converting back to phosphoglycerate, what will happen? One thing is cell is utilizing energy. It is the loss of energy. Another thing, on the way, it will lose carbon dioxide. So the 3 carbon, for example, the 3 carbon dioxide that we need to form glucose, so that glucose will not form again. It means we lost this 3 carbon dioxide. Or we can also say that if this 3 carbon dioxide is made, or if the glucose form, so maybe RUBP, maybe this RUBP will decrease. So one thing is loss of RUBP, another loss of energy. Nothing happened means like we are losing this carbon skeleton. So this process is a bad process. In order to avoid himself from this photorespiration, some plant evolved adaptation, and that are called C4 plants. And what are that adaptation in that plants? The first adaptation is these plants having special enzyme. That enzyme is called phosphoenol pyruvate carboxylase. This enzyme, and PEP, we call that PEP. That is fixing carbon dioxide if oxygen is more or less no matter. And another adaptation they having bundle sheet. Around vascular bundle. These are some cells that are very near to each other, no gap between that. Means like no airspace. That is around vascular bundle, right? So these are what bundle sheet. So how this help? Let's suppose if this is vascular, this is your mesophilic cell, right? So this PEP carboxylase in this mesophilic cell, if oxygen is more, so no matter what will happen, this will convert this carbon dioxide into what? This will convert this carbon dioxide. This will combine this carbon dioxide with PEP. This will combine this carbon dioxide with PEP, phosphoenol pyruvate. Just look toward here. I will discuss this pathway to you here. You will understand it well. You will understand it well, this pathway. Let me select this one. Now look toward here. This carbon dioxide will fix by what? By PEP carboxylase enzyme. Am I clear? And when this is fixing that, it will combine it with what? Phosphoenol pyruvate, with another molecule, phosphoenol pyruvate. Normally it was with ribulose biphosphate carboxylase, but because of this adaptation, PEP carboxylase enzyme adaptation, if oxygen is more, no matter, this carbon dioxide will convert it to what? Oxaloacetate. 4 carbon molecule form. This 4 carbon molecule, because of this, these plants are called C4 plants. C4 plants. This oxaloacetate will now move toward where? To bundle sheath. As I discussed with you, these are bundle sheath. You can check here these cells, right? And these cells, oxygen concentration is less. So this oxaloacetate will convert it to malate, and malate will enter to that bundle sheet where oxygen is less. So if oxygen is less, there is what? Ribulose biphosphate carboxylase enzyme. So this enzyme will combine carbon dioxide with RUBP, is normal dark reaction start. And that will pass through that Kelvin cycle, and again it will form what? RUBP again. And on the way it will do for example, this is 3 carbon dioxide. So these 3 carbon dioxide will give us in the form of sugar or any other molecule, organic molecule. If this is 3 carbon skeleton, so another 3 carbon skeleton combined with it, it can give us glucose, or it can utilize for any other organic molecule, right? If it is sugar, so this will move toward vascular bundle and will transport to plant body through phloem. Am I clear? So here we did nothing, just we take this carbon dioxide from higher concentration of oxygen to lower region of lower concentration of oxygen, so that this ribulose biphosphate carboxylase enzyme does function. C4 plants are like sugarcane, corn, these are C4 plants. Am I clear? In adaptation in that is PEP carboxylase and presence of bundle sheath. So this pyruvate, when malate was converting into pyruvate, it released carbon dioxide, and that carbon dioxide will fix by RUBP. This pyruvate will go back and it will convert into what? Phosphoenol pyruvate. Okay. Today we will discuss about the difference between C3 plants and C4 plants. So leaf structure, we discuss in C3 plants, no distinct bundle sheet. There is no bundle sheet. While in another bundle sheet still having chloroplast present in C4 plant. This is a specific adaptation in C4 plant. C3 plants having robust coenzyme that are doing fixation of carbon dioxide. Am I clear? While C4 plant having what? Pep carboxylase enzyme. C4 plant having what? Pep carboxylase enzyme. Through pep carboxylase, they are doing what? Fixation of carbon dioxide. Optimum temperature for C4 plants is 15 to 25. While for that, they can survive at high temperature, 30 to 40 degrees Celsius. C4, C3 plants occur in the same cell as Kelvin cycle, carbon dioxide fixation. And C3 plant is occur in the same cell, while in C4 plants occur in different cell than Kelvin cycle. First of all, pep carboxylase will fix that carbon dioxide, and then in the form of millet, it will move toward the bundle sheet, and bundle sheet Kelvin cycle will take place. Okay. C3 plants product of carbon dioxide fixation, the first one is phosphoglycerate. That is C3 molecule, 3 carbon molecule. While in C4, that is oxaloacetate. That is what? Oxaloacetate. This is 4 carbon molecule. The first product after fixation of carbon dioxide. C3 plants occur in special character of this occur to most plant this. But C4 plant thick leaves lives in dark and hard, dry and hard climate, and bundle sheet used to prevent water loss. Am I clear? Now, C3 plant example, rice, wheat, grapes, coffee plant, all these are what? C3 plant. And here sugar cane and corn. These are the example of C3. Am I clear?
Dear students, the topic of discussion is photosynthetic pigments. Photosynthetic pigments are pigments present in chloroplast or photosynthetic bacteria that capture light energy necessary for photosynthesis and convert that into chemical energy. Is in light reaction of photosynthesis, sunlight will trapped by chloroplast, and that is converting into what? ATP and NADPH, and that is utilizing in dark reaction of photosynthesis, right? Now, to pigment is actually substance that absorb light. Now, the color of pigment come from the wavelength of light that are reflected. For example, chloroplast reflecting which wavelength of light? It's actually reflecting green light. Because of that, we can see chlorophyll. Chlorophyll is reflecting green light. That's why we can see this pigment what? Green color. While keratin, the reflecting orange wavelength of light. So that's why we can see that what? We can see that orange or yellow color you can see. Okay. Now, if the pigment reflect, for example, if pigment absorb all wavelength of light, that will be black. While if pigment reflect most of the wavelength, that will appear white. The light absorption pattern of pigment is called absorption spectrum. What is absorption spectrum? The light absorption pattern of pigment is called absorption pigment. When pigments absorb light, electrons are temporarily boosted to high energy level. Energized electrons move further from the nuclei of the atom, right? And that will lose energy, that will lose energy. That energy will be in the form of, thermal energy, heat energy, or it will be in the form of fluorescence. You will see fluorescence, right? So that will be in the form of fluorescence or heat energy. And that energized electron will come back, right? Now, you can see here, for example, this is your this is atom, right? So when photon means light fall on that, so electron will get excited, it will move to higher energy level. This is actually
this is actually HM and chloropel A. So when heat fall on that, electron will get excited from that. And now heat, that electron will move to higher energy level, and then that is coming back, it will lose heat, light in the form energy, it will last in the form of light or heat. Okay, now let's see here, photosynthetic pigment in chloroplast. Chloroplast contains several different pigments which absorb different wavelengths of light. The photosynthetic pigments of here found from two groups, chlorophylls and keratinite. Chlorophylls absorb red and blue-violet light. Chlorophyll is absorbing these two types of light, red and blue-violet light, while and giving green, while it is reflecting what? Green light. You can see chlorophyll is absorbing red and blue light, while it is reflecting green light. So because of that green reflection, because of that reflection, we can see the chloroplast green. Why? Because it is not absorbing that light. When it is not absorbing that light, that can fall, that fell and go and fall on your eye, and you can see that what? Green. So leaf pigment absorb most color, right? But it's reflecting green color. All right. Now keratinite. Keratinite, orange pigment that protect chlorophyll from damage by formation of single oxygen atom free radical. They is they can also absorb wavelength of light, and they can also absorb wavelength of light that chlorophyll can't absorb, and pass on some of energy from light to chlorophyll. Actually, keratinite is a necessary pigment that is absorbing light and giving that light to what? Giving that light to chlorophyll A, that is primary pigment, and from that electron will get excited. Now you can see here, in this case, chlorophyll, blue and red color, it's absorbing more, right? While green color, it is reflecting. For keratinite, you can see it's absorbing like blue and green, right? And it is reflecting in this range, yellow or orange. That's why you can see keratinite, what? Orange color. Here, absorption is less at this place. All right. Now, another one we have. So the we should be clear. All right. So there are two types of keratinite, keratin and xanthophyll, right? Beta keratin and xanthophyll, right? Color of beta keratin orange, xanthophyll yellow, and for chlorophyll, chlorophyll A and B, chlorophyll A yellowish green, and chlorophyll B we having blue green. Am I clear? So main and necessary photosynthetic pigment, main is photosynthetic pigment, we have chlorophyll A. This is primary pigment from where electron will get excited. Right, is the most abundant pigment in most plants. Its absorption peak are 430 nanometer and 662 nanometer. It emits an electron when it absorbs light. While accessory pigment chlorophyll B and keratinite, these absorbing light, and that light will transfer to chlorophyll A. And now this is absorption spectrum, right? And action spectrum of chloroplast. So this one is absorption spectrum. Is the absorption spectrum is that spectrum that only show us different it pigment the different pigments, which wavelength of light absorbing by which pigment, right? So that is absorption spectrum, while action spectrum is that spectrum at which wavelength rate of photosynthesis is high. Let's suppose if you having aquatic plant, right? You having aquatic plant, for example, right, in water, and you are exposing that aquatic plant to different wavelength of light. If you are exposing that, so from that you will see that to which you are exposing that to different wavelength of light. Like for example, here I am exposing it to 660 nanometer, 550 nanometer of light, right? So a different wavelength of light, you are checking that at which wavelength of light rate of photosynthesis will be high. So definitely in photosynthesis, oxygen will produce. So if oxygen bubbles are more produced, for example, at 600 nanometer, more oxygen bubble produced. You observe more oxygen bubble in water is compared to 550 nanometer, right? Less bubble you observe. So it means at this wavelength of light rate of photosynthesis high. This is just example. So what is action spectrum? Action spectrum only show us at which wavelength of light rate of photosynthesis is high. So you can see here, this is action spectrum. If this wavelength of light, rate of photosynthesis is high, 650 nanometer, and here at 450 nanometer, rate of photosynthesis is high, where here it is less. And I explain with you how you will do that experiment and how you will know that which just expose plant to different wavelength of light, and 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 find the rate of photosynthesis at which rate it is high. So that is action spectrum, right? Absorption spectrum is the graph showing the percentage of light absorbed by pigment for each wavelength of light, right? While action spectrum of photosynthesis is the graph showing the rate of photosynthesis for each wavelength of light. The rate of photosynthesis will not be the same for every wavelength of light, right? All right, so another topic we have chromatography. Chromatography is actually a technique in which we are separating different substances in a mixture, right, on the basis of their solubility in a solvent, right? For example, we having extract from plant leaf. We having extract from plant leaf. That green extract, that that we isolated from plant leaf having different pigments, right? Now these different pigments we are that contain keratinite, chlorophyll A, and chlorophyll B. So we can separate these three pigments from each other on the basis of solubility in solvent, right? By process of chromatography. So what we have to do in chromatography, the first thing, in chromatography, we having chromatographic paper, right? This is chromatographic paper. So, yeah, chromatographic paper we have. Chromatographic, right? Chromatographic paper we have, right? So this is stationary phase. This chromatographic paper is what stationary phase of chromatography, stationary phase. Okay, on this paper, what we will do, we will make a baseline, we will make through pencil a line, and after that we will put our pigment, the extract, okay, here. So we will mark it. From here we will know that how much distance it is traveling. After that we will take our this chromatographic paper, right, containing pigment, and we will take solvent. We can take alcohol solvent, okay, like ethanol we can take. Is it okay? And we will keep that our chromatographic paper in that. Am I clear? And that chromatographic paper we having what already that extract. Now on the basis of this solubility in this solvent here, we having ethanol solvent, right? So on the basis of solubility of this pigment and the solvent that we are using, so on the basis of that, this pigment will start to move in this through this paper through capillary action through the pores of this paper. So the one that is more soluble in this solvent, that will cover more distance, and the one that is less soluble, it will cover less distance, right? Now if you see here in this example, so this is our baseline, right? From where this is solvent, right? And this is our baseline from where our extract start to separate from each other. So chlorophyll A remain here, right? Sorry, chlorophyll B. Chlorophyll A remain here, right? And keratinite reach to here. It means keratinite was more soluble in this solvent, right? Now what will happen for this each pigment? You have to find RF factor, retention factor. What is retention factor? Retention factor is actually distance traveled by pigment spot and distance traveled by solvent. Distance traveled by pigment spot mean, for example, if I'm considering here for chloro, this is actually the distance traveled by each pigment, right? While distance traveled by solvent is that from baseline up to the last point where the solvent reach, this complete distance. This distance is called what? Distance traveled by solvent. This distance. Am I clear? You can see it here. Let me show you this complete distance. This is called what? Distance traveled by solvent. Okay, distance traveled by solvent. Okay, while distance traveled by pigment spot is distance traveled by each pigment. So for each pigment you will find RF value. So for example, here I'm considering distance traveled by pigment spot, I'm considering chlorophyll A. So this distance covered by chlorophyll A, which distance? This distance covered by chlorophyll A. Let me make it to the, yeah, this distance traveled by chlorophyll A. This distance traveled by chlorophyll A. All right, I hope it is clear up to here. So let's see this one. Okay, now if I'm going to find it, if I'm going to find RF for chlorophyll A, so let's suppose distance travel by solvent, that is for example 10 centimeter, right? Okay, while for example for chlorophyll A, distance travel 2 centimeter. This is just example. So RF value for chlorophyll A, we will have what? 2 divided by 10. Am I clear? So same you can find RF value for what? For chlorophyll B, for keratinite. So for example, for keratinite, distance travel how much? 8 centimeter. So what you will do, you will divide 8 by what? By distance travel by solvent. So total distance travel by solvent is 10. So it will give you value. So for different pigments, you will having different RF factor. RF factor is actually retention factor. Okay, so RF value is in chromatography is distance traveled by given component divided by distance traveled by the solvent. This way will very different on the solvent used, but in general keratinite having RF value close to 1 and chlorophyll B having much lower RF value, and chlorophyll A is an RF value between those of keratinite and chlorophyll B, right? Right, so this video will help you. Just watch this video. This is related with chromatography, how we are separating different pigment, uh, different photosynthetic pigment by the process of chromatography.
Used to separate colored substances, especially pigments.
You can
in paper
here.
chromatography, the sample is distributed between the
This is stationary phase paper, right?
The stationary phase is
We
usually a
having
piece
paper.
of high-quality filter
Yes.
paper. The mobile phase
This is
developing
mobile
solution
phase. It is
that travels
solvent,
up the stationary
right?
phase, carrying
Solvent
the sample
is moving
quickly.
to
When the
stationary
mobile
phase.
moved
On the
over
basis
sample
of
on the
that,
stationary
on the
phase,
basis of
the
solubility in
components of the
that,
sample
it
get
is
gradually
separated.
separated from one another. Our aim here is to separate the pigments from an extract of spinach leaves by paper chromatography. Materials required: chromatographic chamber with lid, extract of spinach leaves, isopropyl alcohol water mixture, Workman filter paper strip, fine capillary tube, thread, scale, and a pencil. Procedure: take a Workman filter paper strip and draw a line with a pencil.
Yeah, we have
about
to make
2
a baseline
centimeters
where
from
we have to
end.
put our
Draw
extract.
another line lengthwise from the center of
Alright.
the paper. Name the point at which the two lines intersect as P. Take the extract of spinach leaves
So
used
now
in
we
a
are
capillary tube.
transferring our
Put a drop of the
extract.
leaf extract at point P.
Alright.
Allow it to dry in open air. Put another drop on the same spot and dry again, so that the spot is rich with the mixture.
Yes,
Take a piece
alright.
of thread and tie the filter paper with it.
Now we are
Suspend the
going
filter
to
paper vertically
suspend
in
our
the
paper
chromatographic
and
chamber containing
solvent.
which is a mixture of isopropyl alcohol
Here solvent
and
we
distilled
use what?
water.
Isopropyl
Make sure that the pencil line
alcohol
remains
plus
about 1
water.
centimeter above the solvent level.
Alright.
Keep the chamber undisturbed for
You
some
can
time.
see solvent
Notice the rising solvent
moving
along with the leaf extract.
along the
As the solvent
paper.
has risen, you will
So
notice different
pigment is
spots of the
separating
pigment,
on the
such as
basis of
keratin,
solubility,
xanthophyll,
xanthophyll, chlorophyll A, and chlorophyll
of
B.
spinach leaf
You can
extract
check
of the filter paper.
keratinite covered more distance.
Take the filter paper out of the
Now
chamber
you
and
have
mark
to
the
find
distance that the
what
solvent has risen
RF
on
for
the
different
paper.
pigments.
This is called the solvent front.
Solvent front, the distance
Dry the filter paper
covered
and
by
measure
the
the distance
solvent.
of the solvent front from the
Alright.
original line and the distance of each spot from the original line.
So by using this we can find what RF vector for each pigment. I hope you can understand it well.
RF is equal to distance traveled by the component from the original line divided by distance traveled by the solvent from the original line. Precautions: always make use of a fine capillary tube. Keep the chamber undisturbed and covered during the experiment.
Right.
The spot should be rich with the sample.
With the sample, it should be rich with the sample.